Precise roles are not clear. That's not all been sorted out yet, although there has been some progress. The proteins are recognised by something called SRP, signal recognition particle, or SecB, as it's called in bacteria, recognising this N-terminal signal sequence up to 20 to 30 amino acids, sometimes even a bit shorter than that, maybe down as low as 12 to 15. This is targeted to the translocase, so the SecB complex binds to SecA. The SRP complex binds to a different receptor, this FTSY. But if we look at the structure of the signal peptide, this is a cartoon form here, we can see in the signal peptide this N region, which is typically polar. There's a methionine and a handful, 2, 3 other residues. Then you get into this H region, which is hydrophobic. That looks in many ways like a transmembrane domain, and that's because it is actually targeted to the membrane. And then there's a C region, a short little tail of a few polar residues, and then there's a cleavage site, and this gets cleaved off. There's a second system which we're going to talk about in a moment called TAT, which has a similar kind of signal peptide, but there is some variation. It has this twin arginine motif in the signal peptide. So we've got this SecY, e.g. pore forming. The energy is provided by SecA, hydrolyzing ATP. The protein motive force also probably plays a role in the translocation of these proteins, driving them through. And as I mentioned before, sometimes you get coupling of the secretion with the elongation at the ribosome, with the protein synthesis itself. That also can help drive things through the system, drive these proteins through the system. So you've got SecB coming in here with SecA, ATP being consumed to generate ADP, and engagement with with this pore and going through. On the periplasmic side, there is a signal peptidase, and that's what actually cleaves off that signal peptide as the protein comes through, as it's finished coming through. And once the protein has then gone into the periplasm, it's then free to fold up, adopt its native fold, and actually become functional. So it has to stay unfolded as it threads its way through the pore, but after that it's free to fold up. So the Sec translocon is the most widely used translocon for the purposes of getting stuff across the cytoplasmic membrane. But in the last 10, maybe even 15 years, there's been a lot of interest in a newly described translocon known as the TAT translocon. It's not quite as widespread. It's found in many bacterial and archaeal inner membranes, found in some plastids, I should say, as well. It takes its name from an abbreviation of twin arginine translocation. And this is, if you look in the signal peptide, you'll find, in the signal sequence there, you find this pair of adjacent arginine residues as a characteristic sequence motif for this system. It's not always required. Sometimes you get lysines instead of arginines, but generally it's that kind of very polar residue that's seen there. Now this is unlike the Sec-dependent system in that it doesn't require proteins to be unfolded. It actually quite remarkably can translocate folded proteins across the membrane. If we're looking at bacteria, we see that there are many proteins that are, let's say most, I borrowed this from Julia, I don't know, she's got most there. But there are many secretory proteins crossing the inner membrane using TAT. The system relies on a complex of TAT-A and TAT-C integrated into the membrane. There is this accessory protein TAT-B, which is found in some bacterial species, but not in all of them. And the energy for translocation here is it's not driven by ATP, instead it's driven by the protein motive force driving things across that membrane there. So here we have the ribosome producing the protein delivered to TAT ABC and going through the system to come through to the other side. So how does it actually work? We have these twin arginines in the signal sequence. They bind, mediating binding to the TAT B C complex, or to TAT C alone where TAT B is not present. TAT A is then recruited and you get this weakening of the membrane and a conformational change in the TAT BC complex, pulling the substrate through the membrane. And you get a proton flux through the hole, which is partly driving the protein through as well. And then the membrane gets sealed up and the substrate gets released from the other side of the TAT BC complex, and then has an encounter with the signal sequence peptidases on the periplasmic side, which cleave off the signal peptide just in the same way as it happens with the SEC system, and your protein is released on the other side of the membrane. Okay, so with systems like SEC and TAT, we get proteins across that cytoplasmic membrane into the periplasm, across the inner membrane as we call it in gram-negatives. Where do they go from there? Well, many proteins will just fold up and have a role in the periplasm. There are at least 100, probably more than 100 proteins that actually inhabit the periplasm in gram-negative bacteria. That's where they have their job. Some of those proteins will actually be targeted into the inner membrane or into the outer membrane, become integral membrane proteins. And many components of secretion systems that are actually involved in secreting things further afield out of the cell start off by using components that rely on the SEC or TAT secretion systems. But if things are going to get outside the cell, we have to have this second step, translocation across the outer membrane. And here are a number of different systems that we can talk about: the chaperone-Usher system, the type 5 secretion system known as autotransporters generally, and type 2 secretion. And so for the rest of the talk, I'm just going to give you a little bit of an overview of each one of those. In later talks, we're going to cover some other secretion systems in a lot more detail, but here we're going to just go through a quick whistle-stop tour of these different systems. Now, chaperone-Usher I already mentioned in a previous lecture, the one on E. coli, so I'm just going to remind you about that one. So if you remember, we said that there was this system where there was a periplasmic chaperone which binds to these pillin subunits while they're in the periplasm, system in the right kind of folding, prevents them interacting with each other, it's pilli from polymerizing in the periplasm. And then there's an outer membrane usher, which acts as a platform for assembly. And these systems are involved in the assembly of different kinds of pili, type 1 pili and packed pili. And as I say, I've already covered this in the lecture on E. coli. So I'm just putting this slide in here as a place marker, just to remind you to kind of integrate your thinking. So that we're talking about systems here. We spoke about an organism earlier, but everything trying to be interconnects. Now, type 5